Sweet. Dr. McNeil, Alex Shaw behind the camera. We're going to talk about termination and transcription in prokaryotes, right? And we got all this information, we being geneticists, got all this information really early on from the study of E. coli. So all this information comes from E. coli. And I'm about ready to tell you the two major ways that E. coli terminates transcription, okay? Row independent occurs in about half of E. coli genes. And row dependent termination transcription occurs in about half of E. coli genes. But these are not the only two ways to terminate transcription. They're just 99% of the way. So that's all we care about. The main things are the plain things. So what do we got? Let's do row independent first. So here's our situation. Double-stranded DNA genome. You see the +1 side comes off of the coding strand. The template strand is the other strand in a 3' to 5' direction that RNA polymerase is reading. Remember, RNA polymerase is a DNA-dependent RNA polymerase because it reads DNA in a 3' to 5' direction and writes new RNA in a 5' to 3' direction. Remember too, because this is prokaryotes, we have our +1 site, our transcription start site. We have part of a DNA that codes for the 5' untranslated region. Here's a 5' UTR. Then we have not exon 1, but gene 1. Okay? Gene 1, gene 2, gene 3, because one transcript in E. coli and many bacteria encode multiple proteins, multiple polypeptides, right? There's no splicing, there's no funny stuff here. It's gene 1, gene 2, gene 3. Okay? Check out translation videos if you want to see how E. coli can adapt to having more than one polypeptide encoded by a single transcript. Even if you don't want to, it's on the testicle form. So what do we got? We have a newly formed transcript. RNA polymerase chunking down, writing new mRNA from 5' to 3'. As it does this, in some genes, there is enough DNA that codes for what we call a stem loop. That was an awful stem loop, I'm going to draw it again. Codes for a stem loop. So as RNA polymerase moves down, what it kicks out, there's a better stem loop, is a mRNA that binds to itself. Why? Well, if you think that this is still the 5' end, you can imagine how this could bind to itself. If we go 5' and let's say UCC, UCC, G, A, U, C, G, C, G, G, A, G, G, A. And now we're in a 3' direction. So from 5' to 3' and 5' to 3', this U binds with the A, it's complementary to itself. C binds to G, C binds to G, U binds to A, C binds to G, C binds to G, G binds to C. So we have a stem, and we have a loop where 4 bases that don't bind to each other are located. So that's our stem and our loop. Okay? Now, that doesn't seem weird, too weird. A lot of RNAs are self-complementary, about 50% of them in E. coli. And so as RNA polymerase moves down, though, what happens? Well, RNA polymerase is bound to another protein, because RNA polymerase is a multi-protein subunit. And one of those proteins is called NUS-A. And the deal with NUS-A is that it binds stem loops. So, as RNA polymerase is flowing down, all of a sudden NUS-A comes into contact with a stem loop, Remember, NASA is part of the RNA polymerase hollow enzyme. The fact that this stem loop is now tugging on NASA from behind, is now dragging, essentially dragging this mRNA, knocks RNA polymerase off. How does it do that? Well, RNA pol falls off because unlike DNA polymerase that has a clamp on it called the beta sliding clamp that holds it steadily onto the DNA, RNA polymerase doesn't have anything. It just chomps down the DNA, writing new RNA. So any little perturbation, like a small stem loop structure giving, let's say, a tug, causes RNA polymerase to fall off. So if you're a steps-oriented individual, 1, you write mRNA. 2, in the 3' UTR, usually exists a stem loop, a stem loop site. I made this one up, but as you can see, it's self-complementary. NASA, which is part of the RNA pol, binds to the stem. And 4, that physical pressure removes RNA pol from the DNA. Simple as that. Totally unique, totally easy to do. Biological wonder. What's going on with Rho? So that was Rho independent. This is Rho dependent. So we're going to utilize a protein called Rho. Well, not we, but the E. coli that hang out in your butt are utilizing it right now. And they're utilizing on about half the genes in E. coli. So again, we have a transcription start site. Transcription gets initiated. Transcribe all the way, 5' UTR. Transcribe through gene 1, gene 2, and gene 3. Transcribe into your 3' UTR. Okay? Which is all the parts after gene 3. What's going on? Well, for Rho-dependent termination of transcription, what you need is what we call a CG island. It's actually about, generally about 80 C's and G's in a row. Why do you need 80 C's and G's? C's and G's all together is the Rho binding site. Okay? So, what's Rho look like? Rho binds to these C's and G's, and Rho, it's going to be in orange here, looks like a horseshoe or a toilet seat. Okay? Rho's really special though. So Rho, in an ATP-dependent manner, burning ATP, utilizing ATP, moves in a 5' to 3' direction down mRNA molecules. So Rho is translocating from left to right. Okay? Here's where it gets trippy though. Rho can only move half the speed of RNA pol. So, how does Rho ever catch RNA pol? Both of them are running to the left. How does RNA pol ever get caught by Rho? Great question. What happens is, usually a few hundred bases downstream of the Rho binding site is a site on the DNA called the pause site. And geneticists a long time ago realized there were pause sites, and there's a couple ways pause sites work. Not super important. One is that RNA polymerase reaches a whole bunch of A's and T's and it starts slipping, and it tries to rewrite the same mRNA over and over and over again in that area so it doesn't physically move down the chromosome. Other RNA polymerases actually get stalled and physically stop moving altogether, but it hits a pause site. So Rho is moving from left to right, and Rho, outside of translocating, so moving from 5' 3' direction, Rho has one important enzymatic activity. Rho is a helicase. And Rho's special job as a helicase is to break RNA DNA hybrid. So Rho is translocating from left to right. RNA polymerase gets stalled where it's at. Rho comes up. All these hydrogen bonds just on the backside of RNA polymerase that form as a result of RNA polymerase using DNA as a template for RNA, uses base pairing to do that, so there's a slight RNA-DNA hybrid molecule for a very short distance. As Rho moves down, it barges in there and starts busting these bonds up, and it busts and busts and busts through these bonds until it's now kind of pulled apart the RNA and the DNA, pulled them apart from each other, and that perturbation in space knocks RNA polymerase off. RNA pol falls off because Rho slammed into it. That's how you terminate transcription. Rho independent, utilizing the stem loop structure. Rho dependent, using Rho, a bacterial helicase.